Right. Good evening. Good morning. Good afternoon. Whatever time it is that you're watching this, welcome. My name is Marco Bauk, and I am a senior here at UCSD, and today I will be presenting this paper. So, genetic evidence for high altitude adaptation in Tibet. That's the name of the paper. The authors, Tatum Simonson, Yingzong Yang, Chedi Huff. Not going to try to pronounce those, but they're there. Those are the other authors. And the central question, what is the central question this paper is addressing? Well, here it is. What is the genetic basis behind Tibetan populations' unique high altitude adaptations? All right, well, what's the significance of this paper? Why does this paper matter? Well, it matters because it's trying to uncover the genotype that correlates with the Tibetan phenotype. So the physiological response of having low red blood cell concentration relative to other high altitude populations and lowlanders that temporarily move to high altitude. So these Tibetans exposed to high altitude, they're responding with relatively low red blood cell concentrations relative to other people. All right, maybe that doesn't make sense. I'm gonna give a little background. Hopefully that'll clear it up. So, like I said, Tibetans are a high altitude population. They live on the largest and highest plateau on Earth, the Tibetan Plateau, hence their name. And being so high up, there's very low oxygen. If you or me were to go up there, we would have an immediate response of increasing how many red blood cells we have in our body because the oxygen is so low. So if we want to maintain normal aerobic metabolism, we need to make more red blood cells to get that lower concentration of oxygen into our body, right? So we may survive. For whatever reason, Tibetans don't respond that way. They don't increase their red blood cell count. So they have very low arterial O2, yet somehow they're maintaining normal aerobic metabolism. So I left for a second. I went to the bathroom, but I'm back. Okay, methods. So the authors of this paper perform a genome-wide scan to identify positive selection in Tibetans. And they did this by, well, first, they looked at genes they knew were putatively associated with high altitude adaptation. So genes that usually, like when you go to high altitude, show varying expression. And then they sampled 31 Tibetans. They were unrelated. They genotyped them for 1 million SNPs using Affymetric genome-wide SNP 6.0 array GWAS. They use GWAS. And the expression of these genes, of these SNPs for the Tibetans, they were compared to Chinese and Japanese lowland populations. And they used XPEHH and IHS tests. If I start talking about it, it'll go over time. But IHS stands for Integrated Haplotype Score, and I think that's going to come up in a second. But regardless, they compared these two populations, so Tibetan highland population to lowland Chinese and Japanese populations. And they were looking for differences in expression. And that's how they were seeking to find out which genes were expressed differently, positively selected in Tibetans, right? So once they had these genes figured out, they were going to, or they weren't going to, they did correlate them with red blood cell concentration. Okay, I took another break, but I'm back. Okay, so here we have the genomic regions of the two populations. Well, technically three, but it's the high altitude Tibetans and the low altitude Chinese and Japanese. Those are kind of paired together. So we have the two genomic sequences compared through the use of these tests. They were able to figure out right here in these black lines. Oh, sorry. In these black lines, we have 10 genes that stood out the most. that were the most difference between the two populations. So here, I'm not going to get into the intricacies of these two tests right here, but all I know is that the authors were able to use these statistical tests to determine out of the 10 genes, right? Out of the 10 genes in here, which two were most significant. Okay, so apparently it was EGLIN1 and PPARA. And here we get this result right here. So what this is 30 Tibetan populations. And we're looking at 2 genes and we're looking at haplotype copies, right? So Tibetan individuals who had none of these haplotype copies associated with the SNP variant, they had the highest red blood cell concentration. Those who had one copy had slightly lower red blood cell concentration, and those who had both haplotypes haplotype copies had the lowest red blood cell concentration, right? Same applies to the PPARA gene. Okay, so EGLIN1 and PPAR are the genes that contain varying haplotypes that are being positively selected for in Tibetans. In summary, that's what this paper is telling us. So context. Well, now the authors know which genes, which SNPs are, which SNPs in the genes are contributing to this low red blood cell concentration expression that these Tibetans get. But I guess context and what other questions they could ask are, okay, so they have lower red blood cell concentration, meaning they also would have low arterial O2, right? So how are they maintaining normal aerobic metabolism? So for example, they could have higher tidal volume, but what are the genes that encode for that? So there are other questions that can be asked. They could delve a lot deeper. This is the tip of the iceberg. And I think there's another presentation that goes into the mechanisms actually of this exact study. It's a following study. So I would look into that one. All right, cool. Thank you guys. I appreciate your time. Good luck the rest of the quarter.